Hello, dear everybody. Greetings to you. Myself, Mr. Karthikeyan, mentor in NEET, Botany and Zoology subjects. So, in this video session, I want to explain to you 2 marks answers from Intermediate Public Examination question paper given in 2023 Botany subject, Andhra Pradesh. So, in part 1, already I have been explained first to 5 2 marks answers. So this is part 2 video. Remaining 5 2 marks answers I shall explain to you. Listen carefully and watch and grasp this. Utilize in exam. First one is conjugation. From the lesson bacteria, this was raised. So what is meant by conjugation in the sense? Exchanging the genetic material from one bacterial cell to another by the direct contact or through conjugation tube is known as conjugation. So two bacterial cells, they will exchange the genetic material, that is known as conjugation. So this was first time discovered in the bacterium is known as E. coli, Escherichia coli, by the scientist Lederberg and Tatum. These both members, they were discovered conjugation process or the method in E. coli, Escherichia coli. That's the answer of this question. Then second one is action spectrum and absorption spectrum. Define these. This question is from photosynthesis lesson. So action spectrum, absorption spectrum. What is action spectrum in the sense? Photosynthesis occurs at different wavelengths, that is known as action spectrum. VIBGYOR, you knew already, rainbow colors. So at a different wavelength, a different rate of photosynthesis has been occurred in plants, that is known as action spectrum. Then absorption spectrum in the sense, these photons from the sunlight will be absorbed by the pigments in a different range, that is known as absorption spectrum. So rate of photosynthesis is action spectrum. The absorption method, the wavelength is known as absorption spectrum of the photons. Then one more, capping and tailing. So what is meant by capping? Nothing but adding unusual nucleotide sequence to the 5' end of hnRNA is known as capping. Then adding adenylate residues to 3' end strand is known as tailing. Capping in the sense at the 5' end, tailing in the sense at the 3' end. Capping in the sense hnRNA, unusual nucleotides, tailing in the sense adenylates residues around 200 to 300 base pairs at the 3' end is known as tailing. This is the answer for that question. Then which microbe is used in starting production and what it may do? So statin in the sense which inhibit the cholesterol synthesis enzyme, which inhibits the enzyme which synthesis cholesterol in the body, that is known as statin. So this statin is nowadays widely utilized in medical field. This is a new technology or the technique. So Monascus purpureus is the microbe used in the production of statin. Monascus purpureus, remember this. Then last one, What are crygenes and what are those features or function in the sense? Crygenes present in a Bt, Bacillus thuringiensis. With the help of Bt cotton is produced. Nothing but invented, Bt cotton, created. So in this Bt cotton, nothing but Bacillus thuringiens, there are three types of genes. Those are known as crygenes. Cry 1AC, cry 1AB, cry 2AB. So here, cry 1AB, this gene, it prevents the attack or harmness of corn borer insect, or worm. Then remaining IAC or 1AC and 2AB, they will prevent the effect or harmness of cotton bollworm. So cotton bollworm harmness prevented by these two crygenes. Corn borer harmness is prevented by this gene. These three together called crygenes present in Bt cotton, Bacillus thuringiensis. I hope everybody you understood. This is part 2. Remaining 5 answers I have explained in part 1. You must go through that video. We shall meet in next video with one more year paper, Intermediate Public Examination, Andhra Pradesh. Thank you everyone. Signing off. This is Karthikeyan, mentor in NEET, Botany and Zoology.